My name is Travis Pettengill, and I am in my final year at the University of Florida pursuing my master's degree. I work in the Orthopaedic's Biomedical Engineering Laboratory under Dr. Kyle Allen. The lab specializes in gait analysis and gait parameters which can be used to diagnose many disease states as well as treatments. Rodent models that we have worked with in the past include arthritis of the knee, arthritis of the spine, and generalized neuropathy which some chemotherapy drugs induce. Whenever possible, we use longitudinal experimental designs because such designs increase the sensitivity of preclinical studies and thereby potentially reduce the number of animals per study. We design our experiments around finding the following: stride length, step width, symmetry, and duty factor. Stride length is simply the distance between successive strikes of the foot and is always parallel to the direction of travel. Step width is always perpendicular to the direction of travel and is the projected distance between right and left strikes. This plot shows time on the horizontal axis and either right or left foot on the vertical, but it is effectively a one-dimensional plot. The orange and blue bars represent time that the right and left foot are planted on the ground for a bipedal animal. When you consider this line to be t equal to 0, symmetry is the time of the foot strike divided by the stride time of the opposite foot. For normal gait, this is 0.5. Duty factor is the stance time of a foot divided by that foot's stride time. My challenge this semester was to create a MATLAB function which reads in many high-speed videos and processes them to generate average spatial and temporal gait parameters. I want to emphasize that this is the sole source of raw data that I rely on to calculate parameters. Because parameter changes in time are typically very small and cannot be seen with the human eye, we use a frame rate of 250 per second, typically capturing 4 seconds of video. We use a mirror to simultaneously film the side image of the rat in the XZ plane and the bottom image in the XY plane. This is a sample image of a single frame that MATLAB creates. I have used color segmentation to find green pixels and assign them to black for each frame of the video. It is now possible to use the find function to locate the lowest part of the rodent's body at the floor, shown with the red line. This next image is a montage of still frames of the rat walking. In this case, the rat is moving from right to left as time increases upward. The red lines highlight the floor on each frame. The red bars at the bottom show space in X that the foot will be on the floor at some time in the video. As you watch the red bars translate upward through time, you see a new plot appear. This is a projection of through time for every frame of the video. The large red objects in this image represent time as well as place that the foot is on the floor. These are steps in X time space. Steps are counted with BW label and verified to have a typical step shape with the region props function. Once the steps are identified, MATLAB will look for each step's foot strike at the bottom of the step marked in blue and the toe off at the top of the step marked in green. There are 2 distinct rows of steps on this plot. Forepaws lead hindpaws in time by about 100 frames. These are the steps marked with yellow triangles. This image is the transpose of the previous image. Time has been placed on the horizontal axis and X position on the vertical. 4 steps are marked with triangles and hind steps with circles. If we zoom in on a small section of this plot and exaggerate in time, you will see it is similar to the plot shown earlier to define symmetry and duty factor. In this case, though, the plot is not divided between right and left feet. It is instead truly two-dimensional, with position increasing upward. It is also evident that this chart was made by a quadruped since there are 2 sets of feet overlapping closely in time. Once the timing for each step is known, I work in the XY plane to identify right and left steps. MATLAB will process the bottom image of the video and isolate the pink color of the paws via color segmentation. For clarity in this image, 4 paws can be offset by 100 pixels. The XY centroids are marked in white. Every step in the video has now been assigned a strike time, toe off time, XY coordinate, and has been categorized as right, left, fore, or hind. The final process that MATLAB will perform is to calculate the spatial and temporal parameters for output to Excel. 2 sheets like this one are created for each video, 1 sheet for fore and 1 for hind. Each row in the file represents a step, and steps are ordered by time of foot strike. Reading left to right, each step has an X coordinate, Y coordinate, strike time, toe off time, and either 1 or -1, which represents right or left. Each step from the third upward also has gait parameters associated: symmetry, duty factor, stride length, and step width. MATLAB also returns median parameters for each video. Duty factor is split into 3 cases: all steps, right steps, and left steps. At this point, MATLAB's video processing is complete until the next gait analysis experiment.